Good morning, everyone, and welcome to the forum at St. Bart's. My name is Peter Thompson. I serve as the Associate Rector for Formation and Liturgy here. Thank you so much for joining us for this discussion on vaccines and public health. We're very fortunate to have two of our own, both of whom are professors at the Icahn School of Medicine at Mount Sinai, Dr. Jamie Ferrara and Dr. Jim Marion. Dr. Ferrara is the Ward Coleman Professor of Cancer Medicine and Director of the Center for Translational Research in the Tisch Cancer Institute. And Dr. Marion is Professor of Gastroenterology. We had a conversation about 3 months ago, which was our first conversation about who is my neighbor during this time of pandemic. We talked about opening back up. Today, we're going to talk about vaccines and public health, and there will be future conversations with these valued members of the St. Bart's community. Thank you, Dr. Ferrara and Dr. Marion, for being with us today.
Thank you.
So Dr. Marion, let's start with you. Can you tell us about how a clinical trial works? Who participates in it? Can anyone participate?
Well, clinical trials are, first of all, start with a question. And the question is always, will this intervention or drug, or in this case, what we're going to be talking about, vaccination, work for the purposes that we want, compared to giving nothing at all? Let's sort of put it out there. And the key when you're asking that question, and in fact, it's built into the very statistical model for modern clinical trials. And I say modern because, the first clinical trial ever that was a controlled trial, meaning you're using an intervention, you have a placebo or a fake, didn't occur until 1956. So this is a relatively recent era. Most people are shocked by that. Is something called the null hypothesis, meaning you go into it assuming that it won't work, meaning there is already built in a certain degree of skepticism about whether or not what you're going to do is going to work. So a clinical trial is basically taking an intervention, giving it to a population of patients who fit certain criteria, and these you can be included in a trial based on these criteria or excluded from a clinical trial based on these criteria. You want to try to get a population of patients who either have a condition, if it's a drug treatment, but with vaccination, obviously, it's going to be all comers. You need to have everybody participating if possible, and then identify patients who are at greater risk. The first phase of a clinical trial is a phase 1 or a safety trial. And when I was a medical student, I participated in many of these phase 1 trials. The joke was medical students and prisoners were populated most of these trials. And it was basically a way to determine whether or not the drug is safe. And that's the number one priority. That's why it's in phase 1. If it's not safe, then and you're giving it to an asymptomatic population, you're running the risk of taking a healthy person and making them an unhealthy person because of that intervention. The phase 2 aspect of it starts to get into more, well, what are the pharmacokinetics, meaning how does it interact with the body, and does it achieve the goals that you're looking for that may help in this situation? And for a vaccine, it's, do they generate antibodies? Do they prevent carriage? Do they prevent reinfection? And that sort of thing. The third is, does it really work? Does it really prevent the disease? And in this case, it's a complicated question because it's not just preventing infection, you're preventing a disease. I mean, a COVID, we use the term syndrome because it appears to be a variety of different reactions depending on the patient to this viral infection. So yeah, going in with some skepticism, although my preferred term is equipoise, meaning we don't know when we start these trials. We try to model it. We try to predict how many patients need to be enrolled, what the effect will be. Like a lot of people are probably hearing that the vaccines have to be at least 50% effective, which is on par with our influenza vaccines that we get every year, about 50 or 60% on average, sometimes a little lower, sometimes a bit better. And people are disturbed by that. But in fact, when you start thinking about herd immunity, that kind of thinking actually can be protective for our population. But equipoise is, I don't know yet. Let's see the data.
Got it. Dr. Ferrara, can you talk a little bit about safety in a vaccine trial? Is it common to stop a clinical trial for safety reasons? And I know that there was a trial that was recently put on pause, one of these vaccines.
Great. Terrific question. Yes, it's very common to stop trials. In fact, most trials are written with what we call stopping rules, that we're going to look at a certain number of patients, and if a certain number of them or a certain percentage of them have an adverse event, we will actually, usually it's called pausing the trial. We will stop accrual, we will evaluate the adverse events, and then we will see whether there is a safety signal or not. So this is indeed very common, and the fact that the AstraZeneca trial was stopped for one patient who had apparently a serious neurologic event, where, you know, some people have talked about transverse myelitis that hasn't been confirmed, but that's a possibility that can cause paralysis. That would be a very serious adverse event. In fact, these adverse events are usually defined before the trial starts. For example, an adverse event could be fever. That would be an expected adverse event, and that would not stop a trial. But neurologic symptoms, particularly serious neurologic symptoms, that could, and particularly if they are unexpected, there are time limits that if you have a serious unexpected adverse event, you are supposed to notify the central authorities within 48 hours, specifically so that if needed, the trial can be paused, the data can be analyzed, and then the independent Data Safety Monitoring Board, who doesn't know anything about the specifics of who got the placebo or who got, if it's a phase 3 trial, who got the placebo or who got the active vaccination, they will look and see whether it is probable that whatever the adverse event is related to the intervention. And then if it's not probable, you can proceed. If it is probable, then sometimes they have to do even more analysis to decide whether it's safe. But this is, I think we should actually be reassured by the fact that that trial stopped for one patient. By the way, it's worth acknowledging that COVID itself may cause some of these autoimmune type problems. One patient earlier on apparently had multiple sclerosis or a multiple sclerosis type syndrome. Well, that's an autoimmune disease, and sometimes vaccines can cause autoimmune phenomena or the intensification of autoimmune phenomena. So a very mild case that might not have been diagnosed might now become more evident or serious. So that's usually how safety is determined, but it is often, and we'll maybe talk about this later, within a fixed period of time. Usually the time of intensive monitoring of all of the patients on a trial.
So Dr. Marion, once you have shown safety of a particular vaccine, how do you measure efficacy? How do you know that it's effective?
Yeah, so here is where you really start getting into the complexity of what we're facing here. So just to give you an idea of how concerted an effort globally this anti-COVID vaccine initiative is going, we have, I think at last count, it's over 200 candidate vaccines that are now that are at least in phase 1. There are 3 that have been given sort of, I'll call it, yellow light, the sense that they have already shown very good data in phase 1 and phase 2 and are now already in phase 3. And so those 3 are really the focus at this point. But I really want to emphasize that they may not end up being the final answer. 90% of all clinical trials end in failure, right? Now, you think, my gosh, think about that for a second, Peter. If your sermons only worked in 10% of us, we'd be a mess, right? It's very difficult. If only 10% of the treatments that Jamie and I used on our patients worked, my heavens, we'd have to close up shop. Clinical trials aren't the same as treating patients or using something that's already been proven either by experience or in clinical trial. So the difficulty here is not that we do clinical trials poorly or that we don't have a good understanding. I think we're uniquely positioned with the COVID-19 for understanding at the starting gate than I think we ever have been. And a lot of that has to do with the fact that in 2003, we had sort of, I'll call it coronavirus 1. And that sort of prepared us. There was a lot of work. Beijing really was hit hard by that, but because of the way the infection played out and the way that the world was shaped in those days in terms of people traveling back and forth was far less from that part of the world. The other, a lot of people may or may not remember the Middle Eastern respiratory syndrome, the MERS virus. That is also a coronavirus. And that prompted another round of investigations for developing vaccines. So there was a lot of work already done on figuring out this coronavirus. The other thing that's changed is we have now had a kind of and Jamie as a basic scientist could probably elaborate on this more intelligently than I can, but we've had a real, I think, leap forward in our ability to develop vaccines using DNA technology that didn't exist 20 years ago or even 10 years ago. A lot of our viewers may be familiar with maybe a different vaccine their doctor's offering them for the flu or for pneumonia or for shingles, for example. All of these newer vaccines end up working much better than their previous iterations because of some of these new technologies. All of this has been brought to bear in this setting. And we're looking at, I mean, it is truly astounding how we even got to have 3 vaccines that are now enrolling in phase 3 clinical trials at this point. So I have a lot of optimism, but again, the word here is equipoise. Knowing that 90% of all of these fail for one reason or another. But we've got 200 candidates, we have 3 strong candidates, and I think the idea here is let's look at the data and see what it brings.
Dr. Ferrara, Dr. Marion spoke before about the 3 different phases of a trial. Can you speak a little bit more about why we have different phases in a clinical trial of a vaccine?
Sure. So these phases are designed to put as few people at risk as possible and still get to the answer that we want. So the phase 1 is strictly toxicity and trying to find a dose. Phase 2 is, do we think it works? And do we have an approximate idea how well it works? And then phase 3, as Jim mentioned, is the real proof of the pudding. Now we have a strong idea of how well it works. We know how many people we have to look at to be able to determine statistically that this is indeed sound. And we know that it is safe, although there can still be stopping rules. So as we've seen in the phase 3 trial, if you have a very rare problem, 1 in 10,000 people, or 1 in 100,000 people, you're not likely to see it even in a trial of several thousand, right? So you have to be able to still, if you see a rare event that suddenly is much more common than you would have expected, you still have to be ready to stop. But I've read one of the, for example, the informed consent of one of the current trials, And there, the placebo is actually a vaccine that's already been approved, a vaccine against a bacterium that is not expected to give any protection against a virus. But it might give you the sort of mild problems, fever or soreness, or even a flu-like symptom that you might have after a vaccination. And so I thought that was a very smart way of doing it. But what one also has to remember, and we can probably talk about this later if needed, what is the end point of the trial? The end point is the number of events that you're counting that's going to say it is, let's say the vaccine is protective or not. Well, what's likely the case is in these kinds of trials, a vaccination trial, do you produce antibodies to the vaccine? That is actually, it's going to be a blood test that is the primary endpoint of the trial. And you may be able to say for sure that people produce antibodies. But then there are other questions on top of that. Are these antibodies neutralizing? Well, they may be able to do that with enough patients. And of course, what we really want to know is, does the vaccine protect you from disease or from serious disease? And for that, you often have to wait 6 months before actually determining whether that endpoint has been reached. Now, that may not be the primary endpoint. The primary endpoint may be the blood test, whether you've made antibodies, but still that's important information that you want to know. So even these trials that are ongoing now, that are nearing some of their accrual goals, that sort of answer about whether it actually prevents the actual disease or reduces the severity of the disease, we may not know that for months, even though the trial has reached its primary endpoint.
And do all of these vaccine trials have the same endpoint, or are there different endpoints, and how do we compare them?
Yes, so they don't have the same, my understanding is they don't have the same endpoint. And in some ways that's not good for us, because we'll want to know, it would be natural to ask, does vaccine A work better than vaccine B? You know, are 80% protected versus 50%? But that's not actually the way the trials are constructed. So it's not a completely fair answer. Now, some people have suggested that the government could say, well, look, please use standard measures of antibody test, or how you're going to determine whether the disease is prevented or not. But that has traditionally not been the way the FDA works. They don't say you must run the trial this way. The company comes with the design, the FDA analyzes it, and then says, yes, we agree with this. Here are the statistical plans that you need to use to develop it. But it doesn't say company A is doing it this way, and so company B also has to do it that way. So we won't have that kind of answer, nor, to be honest, nor should we, because the trials weren't designed that way.
So it won't be easy to compare them against one another necessarily.
That's right. That's right. And anybody who says that they can do that, you would really want to have a hard look at the data.
And I would add to what Jamie is talking about here is, while the endpoints are different, I think before I'd mentioned the degree of collaboration that we're seeing here amongst these various companies. They each like to have it first. There's real incentive for that. But their commitment to producing a safe and efficacious vaccine, they've made clear in more recent statements, particularly among the larger companies. So that's pretty unprecedented at this point. Most of these companies hold their cards pretty close to their sleeves in terms of drug development. And the idea here is to be as transparent as possible and to keep and really let the scientific process unfold. Because if you don't do that, and you have it influenced either by commercial or political interests, you can end up getting into real trouble. You don't want to have a vaccine out there that doesn't work, because that will harm people. You don't want to have a vaccine out there that's not safe. That will harm people. And the idea here is that you want to reduce the risk as much as possible.
Well, related to that point, there has been a concern that the vaccine process will be influenced because of political reasons heading into the 2020 election. How can we know that a vaccine is safe and effective when the FDA is being doubted, for instance?
Yeah, that's a very good question, Peter. And I know Jamie will probably have his own answer, but I want to tell you the history on this. When you mix politics with vaccine development in this country, at least, has been a poor one. And in fact, I would argue it has led to an enormous amount of distrust. And you have to go back to the 1970s for that. In mid-1970s, Gerald Ford was president. Now, many of you may remember Gerald Ford was president because his predecessor had resigned in disgrace. So the level of confidence in the United States government and institutions at that point was at a similarly low period. There was a swine flu epidemic that was their COVID, if you will. And Ford really very strenuously pushed for the vaccine development to be moved along to be sort of a victory. Remember, his was a relatively short term because of the resignation. That swine flu vaccine ended up producing Guillain-Barré syndrome and among some of the recipients, and really, frankly, didn't work very well. And that moment in our history is problematic because it really was the seed, the earliest seed of what is now a rather militant anti-vaccination movement in this country. So I think we need to remember that chapter as we're going through this. And I think any predictions, and I'll list a couple of the predictions that I've heard, and I won't attribute them. A vaccine out of Russia that will work for 2 years. Well, you can't say that. 2 years haven't passed yet. This will all go away by Easter, or it's going to be done by October. Well, even the most well-populated clinical trial now, they only have 23 out of 30,000 participants so far involved. Now, they could stop it early if it ends up showing real efficacy. Again, equipoise is the key word here. But to start making predictions without data, you really start getting into trouble. And if you start really fervently hoping that something is going to work, as opposed to simply waiting and seeing, that impatience can be really quite dangerous for patients. And it can corrode faith in these institutions that are supposed to be protecting us.
Yes, I would agree with that. You know, one of the very sad outcomes of the current situation is that the reputation of the CDC and of the FDA, it's been tarnished. There is no doubt that there have been mistakes, and there seems to have been a fair amount of political pressure brought to bear. In today's New York Times, there's a discussion of the call of Francis Collins, the NIH director, and Peter Marks, one of the senior epidemiologists in the FDA, to the White House about the plasma therapy. Now, Jim and I have both been involved with some of the studies here at Mount Sinai, and my impression is that plasma therapy is indeed has some real potential, but it's not perfect, and we don't know how to use it yet most effectively. And part of the difficulty of pushing it to say, okay, it's now therapeutically approved, is that the clinical trials then stop or slow down dramatically, and you don't get an answer. And we've already heard this from a number of different places that we really don't know how best to use a therapy that probably has some side effects and that may not work for everybody. And of course, that's what you want to know is how to do that. So I think that the short answer to the question is, now that the medical community, American medical community at large, is on high alert that there are political pressures involved in this, that we will all demand to see the data and demand to see increased transparency. We're not going to take the FDA's word for it that, oh, it's fine. It's like, okay, let's show us, prove it to us. You know, they have the data, or they will have the data, and we will all be able to evaluate it. And I think that people will probably make up their minds when they get a consensus opinion from the medical community that indeed a vaccine is safe, is effective. It's not going to come from a press announcement from some government agency.
Dr. Marion mentioned the anti-vaccination movement, and one of the concerns of the movement is that vaccines cause autism. How can we prove that that's not the case in the case of a COVID vaccine, for example, when autism is more of a long-term side effect? How would we be able to measure that in the context of a clinical trial?
Okay, so first, let's be clear. Vaccines do not cause autism. that is one of the terrible outcomes of this lack of faith in our public institutions. And it got, you know, it's one of those, autism is actually fairly poorly defined. It's never been clear in even in the deep scientific literature about where it is, but people want to have a reason that their kid isn't normal, and so it just has poured gasoline on brush fires that are willing to start. So if you remember anything else, autism is not caused by vaccines. That is very clear. Now, I can almost guarantee you that years from now, someone will say, my Johnny got the vaccine and he's autistic now, and if he hadn't had the vaccine, that wouldn't happen. That's just not the case. It's going to be, people are probably putting in safeguards to try and look at those data and do long-term follow-up, because it often takes years for that kind of, for autism to develop in people. It's not immediately apparent, and there's a whole spectrum of cases. But if someone comes to you and says this vaccine causes autism, the answer is vaccines have never been shown to cause autism, and this one is not likely to do it either.
Yeah, I definitely want to echo that. This is it's a trope that's been going on for a while. The studies are very, very clear that they do not. And in fact, studies that were published in the Lancet that purported to link them have since been withdrawn and judged fraudulent in retrospect. And investigators involved in those studies are no longer able to practice in clinical trials. The idea here, Peter, I think, if I'm going to sort of convey anything to anybody watching here, is a phrase we often hear from the pulpit is nothing is promised, right? The idea that you can try to predict as much as possible with these studies is really the fervent hope of everybody involved in these trials. We want to try to predict any eventuality we can, but we have to keep our minds open to the greater risk that these particular infections are causing. It has devastated our economy. It has completely shut down our society. It has isolated huge swaths of our population. So the risk and the price of this COVID is extraordinarily high. Even with that, though, I think the precautions that are being made here are remarkable, and the transparency I do have faith in. I have faith in it, but I want it verified, as Jamie said. We're all going to be keeping our eyes wide open. We're not going to take anything on blind faith. And I think for us going forward, we need to keep our minds open as to both the benefits and the risks. You know, for example, you brought up the whole autism and thimerosal-based vaccines. that dogma that they were linked somehow really probably delayed our understanding of exactly why autism was being picked up more frequently, particularly in California. And, it took a couple of investigators looking at it saying, oh, well, the paternal age is higher in these patients. Well, that might be something there. Well, that line of inquiry and thought was lost for a decade because they were down this rabbit hole about something that was completely unrelated. So we miss things. We end up not learning things that we could really use learning about, and knowledge that we could probably use going forward when we close our minds down with these sort of dogmatic pronouncements linking this with that. And I hope that we don't close down the inquiry for finding a vaccine based on that kind of dogmatism.
I encourage those of you who are watching, who have questions for Dr. Ferrara and Dr. Marion, to send them in using the live chat function on YouTube, the comments function on Facebook, or you can simply email me, pthompson@stbarts.org, pthompson@stbarts.org. Before we head to those questions, and we have some coming in already, can we talk a little bit about whether a vaccine will work for everybody equally when it comes out? How might there be differences in different populations in terms of the efficacy of a vaccine?
Well, let me start by saying, when we were talking earlier about who would get the vaccine, typically the volunteers are between 18 years old and, let's say, 60 years old. Well, if you haven't vaccinated people older than 60 or younger than 18, you don't actually know that it's going to work in that population. Now, we just read this morning that one of the trials has expanded its size from 30,000 to 44,000, and that they are lowering the age, so we'll get more information about young people. And they're also including people who have pre-existing conditions, for example, hepatitis or diabetes. And you want to have, so this is great news, actually, because if more people wind up getting the vaccine and they are closely monitored, then we will have a better idea of whether it works in all populations or not. If you see wide variability, let's say that it doesn't work as well in young people, then you might say, okay, well, maybe we need to be careful about vaccinating younger people or do an additional trial or segment of a trial to include them. I think this is going to be particularly important for older people like myself, because the immune system as we age actually develops. It doesn't sin because it doesn't have a conscience, but it does things that it ought not to do, and it doesn't do things that it ought to do as we get older. That's in part how you get autoimmune diseases in older people. So it's not always clear that a vaccine is going to work as well in older people. In fact, in general, it probably doesn't work quite as well. But we'll just have to see what the data show, and if it across groups, if it works relatively well, then it's fair to extrapolate to believe that it's likely to work well in people who actually weren't tested during the vaccine. But that's how we would think about it.
Yeah, and I would echo that further. And again, it is encouraging that they've expanded it to 44,000 and are widening the inclusion criteria for patients based on age. That speaks very well to the safety so far. And I think that, I agree, we should definitely take that as encouraging. The other piece of this is that we do know that COVID itself behaves very differently in different patient populations. And some of the things that we've learned have been quite paradoxical. And again, the idea that age is a risk factor for this, and Jamie, I consider you ageless, but I think the risk that we've seen in terms of age, the risk that we've seen in terms of obesity, in terms of diabetes and hypertension. There were all clues in all of these things. In some of our patients, for example, I work primarily in inflammatory bowel disease. We have learned that, in fact, patients with inflammatory bowel disease, which is an immune-mediated condition, and often these patients are on immune modulators, if they're not on steroids and not otherwise ill from their Crohn's disease or ulcerative colitis, their risk is roughly the same as the general population. And if they're on some of the medications that we use, such as ustekinumab or infliximab, which are, in my mind, immune modulators, their outcomes tend to be a bit better, to the point where there are actually clinical trials using these medications for later stage COVID syndrome. So I think the way that the immunization is going to work on a given population is going to be as complex an answer as the number of patients who are presenting with it. This is we see very unique responses, and we don't understand all of the risk factors, but some of the risk factors that have come out have been really informative. And again, I want to tell you, the first COVID patient I treated was the last week of March, and it's just barely 6 months later, and we've learned an immense amount. So yeah, lots of answers coming, I hope.
Yes, and just to underscore that point, we had the same concern in cancer patients who are famously immunocompromised because of the chemotherapy, and in fact, many of them did either they didn't do worse, and some groups actually did better than we had expected. So we do not have all of the answers yet, and people who make sort of broad sweeping generalizations about, well, this won't work or that does work. Let's take it with some consideration and just some humility that we don't have all of the answers. As Jim says, we've learned just a ton in the last 6 months, more honestly than I thought possible, in part because the scientific community has really come together to focus on this in a way that most of us have found extraordinary, that there's so many really, really smart people working on this from thousands of different angles. But when we come to some answers, let's just make sure that we're being reasonably cautious about how we generalize some of these and not make blanket statements too quickly.
Well, we have a lot of questions. I'll try to get to as many as possible. Again, if your question isn't answered today, we hope to have another forum on COVID sometime in the next few weeks or months. So we hope we'll have a chance to answer your question then. The first question has to do with immune responses to COVID-19 and what we know so far, both post-infection immunity, but also theoretical vaccine-derived immunity. How might those immune responses be different, and how might we be able to measure immunity to COVID-19 in the future?
The difficulty now with natural, I'm going to answer this with 3 parts, and Jamie, chime in whenever you wish. Again, I defer to your basic science knowledge on immunology. There are 3 issues here. One is naturally acquired, meaning you picked it up naturally as an infection. We know that there are a certain proportion of patients who do get COVID. They are swab positive, and they just don't generate the antibody response that you would expect. Again, we don't know enough yet about why that is. It doesn't necessarily track with incapability. It can sometimes track with age somewhat, older or younger. And it also tracks with dose response. Now, remember, I think I mentioned the last time we met on this that the virus has a dose response toxicity curve, if you will, meaning the more you're exposed to it, the more likely it is you're going to have a serious complication from it. And this is why you see ventilated rooms. This is why PPE was so important when we were first starting this. And So it's going to be dose related and it's going to be host related, meaning the patient and where they're at. Similarly, what we're trying to do with the vaccine is we're trying to gauge what is a safe dose of, whether it's a live virus or a protein that is designed to trigger an immune response. Again, we're going to learn a lot from these studies, who mounts a better immune response than others. The question and then finally, going back to the first one, the dose response issue, you may have seen some articles recently looking at masks as somehow providing sort of a soft vaccine for some patients, meaning a lower dose that's acquired naturally for particularly younger patients might be a way for them to generate an immune response. Now, I consider that wishful thinking, and it's not controlled by any measure. How long will it last? Well, the hope is that if you get infected, you're going to be protected. But I will tell you, we were hit quite hard at Sinai, and many of my colleagues were infected, and we lost several. Those who were infected and did mount an immune response were very dutiful about donating their plasma, but they're still wearing masks. And they're wearing masks because we don't know yet whether or not they are protected going forward. And we aren't sure how long. And so far, because we're only a few months into this, the best number we can give for duration of the antibody response is about 3 or 4 months, because that's about how long we've been measuring it. For the clinical trials with the vaccines, it's going to be a similar problem. How long is it going to last? Well, we're going to have, how long is it going to last and protect you? We're going to have to wait a bit. Disturbingly, we are hearing reports here and there of reinfections in patients. And one sort of hopeful sign with the reinfections is that they appear to be milder than the initial presentation. And that may tell you something a little bit about how this virus is going to behave and how it may linger in our futures going forward. It will linger in our future going forward. Again, I think I try to emphasize this as much as possible. This is an historic moment to have this virus go from an animal reservoir into a human reservoir. This is not going to be like smallpox, where we eradicate it completely, or like polio, where we've almost done that, where it's really the human reservoir that's the issue. This lives in an animal reservoir. So we are going to have to figure out a way to control this. We're going to have to figure out a way to manage it in the population, similar to the way that we've had to manage Ebola, right? So Ebola, also animal reservoir. If we don't get vaccines in, we don't figure out a way to control it and mitigate any sort of penetration into the human population, then this could happen again.
And just briefly to underscore this, a colleague of mine who works just a couple doors down actually got COVID in March and wound up being in one of the studies, and they are updating them. And he has high titers of neutralizing antibodies 5 months after being infected. And he said, at least his read of the data is that 4 months, which is as far as they've really got good data for, it's still maintaining. So it's early, but 4 months, 5 months, 6 months, that would be very hopeful in terms of, and that's natural immunity. And at least some of the vaccine studies, mostly in models, have also shown up to 4, 5, and 6 months high titers of neutralizing antibodies. And that's how it's measured.
Some good news.
Yeah.
So what can we learn from a failed trial of a vaccine? Is there valuable information we can learn from a failed trial?
Oh, yes, yes, yes. We, Jamie and I are both old enough to remember when failed studies, failed clinical trials never made it to the literature, right? They were never seen. And that has now changed. You will now, anything that can be learned from a failed trial, and again, failed is not the word I would use, even a trial that doesn't reach their endpoints pushes back the border of ignorance about that topic. And that's really the only point of doing a clinical trial. You really want to push back the borders of ignorance and expand our knowledge base. And a negative trial in this setting, this is a novel area, is valuable understanding.
100% correct. And what's important here is when we say a trial fails, usually that means it did not meet its primary endpoint. It did not reject the null hypothesis, as Jim said. But trials this large, where there are thousands of patients, we are likely to get valuable clues as to where there might be soft pockets or harder pockets or different ways of looking at the problem. So a quote unquote failed trial is very likely to give us important information, and we will ask to see it.
Can you talk a little bit about incentives to create a safe and effective vaccine, and to what extent incentives are helpful and to what extent they are not helpful?
I mean, in terms of enrollment?
I think in terms of the companies that are creating the vaccines. So the money, the kind of adulation, why would a company want to be involved in this process, and are those incentives helpful?
Oh, well, so there are two questions that you just had. For the company, the incentive is billions of dollars, right? They're going to make, if a successful vaccine will guarantee a company's bottom line for years. I thought you were asking, the question was about individuals. Should individuals be incentivized, perhaps even to take a vaccine once it has been shown to be effective? There was something, maybe a podcast, someone recommending that the entire U.S. population be incentivized to take a vaccine, to give them $1,000 to get the vaccine that works, that's been shown to work. And we had a little discussion about this at the breakfast table this morning. I said, $1,000, that sounds like too much. Maybe 100 would be okay. And Flora said, no, I wouldn't do it for 100. It would have to be at least 500. So we all have our price. We all have our price.
I think just to sort of follow up on that, I am asked a lot. In fact, I have two informed consent sitting on my desk offering me a vaccine spot on two of the vaccine trials. And I have to say, I'm often asked by my patients, would you take the vaccine? And the answer is yes, I will take it in a minute if it's proved to be safe and effective. I would have no hesitation whatsoever. And in fact, as somebody who'd been on the front line, I'm apparently, I'm told that we're going to have access to it. In terms of participating in a clinical trial, that was a different conversation. That was a conversation with my wife. That's a conversation with my kids. And for now, I'm letting them sit on I'm not going to let them collect dust for too long, but I'm still thinking about whether or not I'll enroll in the trials. My first attempt at getting naturally vaccinated on the COVID units was unsuccessful. I didn't get it, but so I'm still thinking about it.
All right, we need to stop, but I'll sneak in one last question. All but 5 states still allow folks to exempt themselves from vaccination on religious and belief grounds. Is that wise policy, given that we're living in the midst of a pandemic?
No, it's not policy.
Yeah, it's yeah, I struggle with this because I know that there are really serious political issues surrounding this, and I struggle with any sort of butting up against between the government, and I'll say that with a big G with quotes around it, and religious organizations in this country. It is a minefield area. But I have to agree with Jamie. I think the tradition of having deep suspicion about vaccinations, particularly among religious groups, is not a great part of our religious history. And I'm going back to the 18th century and smallpox and cowpox and all the vaccines that were developed. If you look at that history carefully, religious organizations didn't necessarily play a good role in what we now know in retrospect were not only safe, but saved lives. And I think if we are going to consider ourselves truly pro-life, as it were, that we've lost nearly 200,000 Americans to this. And the illness, we still haven't counted. I mean, there are going to be long-term ramifications for many of my patients who did get it. I think that I'll quote a rabbi who I've worked with for years, whose congregation initially was very resistant about some of our Crohn's disease and ulcerative colitis trials, when he said, it's better if we can prevent the illness in the future. It's almost as good as a cure. And I think that we need to be thinking in those ways about this, and maybe less reactively as people of faith.
Well, we really do need to stop. Unfortunately, there are a lot of additional questions. We will have another one of these sessions in the upcoming weeks and months, as I mentioned. So if you have a great question, hold on to it. We'll address it later on. Thank you, Dr. Ferrara, Dr. Marion, for joining us again this morning. We are so fortunate to have folks with your expertise in our midst, and I'm really grateful for your generosity of your time this morning.
Could I have one last word?
Yes, please.
Go get your flu vaccine. Everybody, all adults, even if you've not had one before. The risk of a twindemic is real. You can protect yourself and really help all of us by getting a flu vaccine. Now is the perfect time to get it. Everybody should go out and vote, and everybody should also go out and get a flu vaccine before they have voted. Please.
And wear a mask.
Thank you both. Thank you all for watching. Stick around and join us for worship at 11 a.m. Thanks a lot.
Thank you, Peter. Thank you.